First of all, I would like to start by welcoming everybody to this special thesis defense this afternoon. And to start this, I will officially open this academic ceremony in which Ms. Sai Sundararaman, it's a long name, will defend her academic titles entitled The Role of PCSK9 and CaSR in Cardiovascular Disorders. Ms. Candidate, may I invite you to present a summary of your studies and the conclusion of your thesis for us in the next 15 minutes, and I'd like to give you the word.
Thank you, Prorector. Dear committee, dear supervisors, dear friends and family, first of all, I would like to thank you all for taking the time to be here and to support me. It's hard to believe that it's been 3 years since I finished all my experiments, and then I started a new job, and I was working on finishing my PhD during my evenings and my weekends, and therefore I've been waiting for this day for a very long time. Now that the day is here, I have to say I'm a bit nervous. But I'm mostly glad that I have my friends, my family, my new colleagues, my old colleagues, all in one room or watching online, and that I finally get the opportunity to explain to you what I've been doing during my PhD. And I'm excited to take you through my research, which is titled Role of PCSK9 and CaSR in Cardiovascular Disorders. Now, you must be wondering, what is PCSK9, what is CaSR, and what are all these acronyms standing for? But before getting there, let's first look at what cardiovascular disorders mean. And as the name suggests, they are diseases of blood vessels and heart, and that includes heart attacks, high blood pressure, heart failure, etc. They are linked to genetic factors, but more often they are linked to poor diet, inactivity, smoking, stress, and aging. It's important that we study them because they are the #1 cause of death worldwide, and they stay silent often until something serious happens clinically, and which can then lead to high treatment costs in terms of medication, surgery, hospital stays, and that decreases the quality of life. So we need to study them in order to prevent them. Now, in this thesis, we are going to focus on atherosclerosis. Atherosclerosis is the main root cause of cardiovascular disorders. It's a condition where fat, cholesterol, and other substances build up inside the walls of the arteries, causing inflammation, and over time, these arteries can narrow and make it harder for the blood to flow. And these buildups are called atherosclerotic plaques. Atherosclerosis is like a messy team project. It has so many players. It has fat cells, immune cells, the cells from blood vessels, all working together. And it's not just one problem. There is inflammation that's causing trouble behind the scenes. The cells inside your arteries keep changing the game plan. And of course, there are so many different players that keep interacting with each other. And it takes years for this slow drama to unfold and making it very hard to predict and fix. It's kind of like solving a puzzle that has moving pieces or missing pieces. And the current treatments that are available don't fully solve the problem, and medications can cause side effects. Therefore, and also we don't understand all causes of this disease. Therefore, in this thesis, we decided to look at 2 special proteins in our body called PCSK9 and CaSR that are known to have important roles in our body and have been rapidly gaining interest in the field of cardiology. So we wanted to shine a new light on them and understand what their role is specifically to atherosclerosis. When we talked about the processes in atherosclerosis, there are so many, and they are so complex, and some of them are highlighted here. And for the first part of this thesis, we want to focus on one important process, which is inflammation. Similarly, there are other processes, and we have already known that PCSK9 plays a role in cholesterol buildup. PCSK9 is short for the proprotein convertase cytosine kexin and is the 9th member of this family. And to understand how it works, let's imagine a busy restaurant kitchen. Normally, you have trash cans that are emptied and reused to keep the kitchen clean. But PCSK9 is like a sneaky worker who steals these trash cans and destroys them. And therefore, over time, the kitchen has fewer trash cans and garbage piles up everywhere. So our cells also have a trash can called cholesterol receptor. This cholesterol receptor is usually present mainly in the liver cells. It recognizes the cholesterol in our body and it takes them inside the cell to destroy or to reuse. But after this process, these trash cans usually come back to the surface of the cell to continue the process. But in presence of PCSK9, these trash cans, or the cholesterol receptors, are destroyed, which means there are fewer trash cans available on our cells, and which then means there is more cholesterol building up inside our body. Now, knowing this, we wanted to see if PCSK9 affects inflammation through other receptors that are present in our body. Now, apart from such cholesterol receptor, there are so many other receptors. Some of them are involved in inflammation. And so our question was to see if PCSK9 affects cardiovascular disorders through other receptors. For that, we looked at chemokine receptors. Chemokine receptors are the cell surface receptors, so they're like antennas on the cell, and they detect signals that are chemokines, and they guide the immune responses and communication between cells. This can then cause inflammation and other key processes in atherosclerosis. So the question here was if PCSK9 affects chemokine receptors. There are several types of chemokine receptors, and out of those, we chose 4 chemokine receptors that are very important in atherosclerosis and checked if their amounts are different in the presence of PCSK9. To do that, we started with mice models. So we wanted to see what excess PCSK9 does to mice. For that, we took mice, we injected the mice with PCSK9, and after 1 week, we saw that these mice had more inflammation. And then we took out the blood, spleen, immune cells, and saw that there was a small change in 2 different chemokine receptors in blood and spleen, respectively. Next, we looked at human cells. For that, we took out immune cells from blood and the cells from blood vessels, grew them in the lab, and put PCSK9 on them, and we saw that the inflammation was upregulated in some cells, and we also saw a small change in 1 chemokine receptor in the cells from blood vessels. So, summarizing the first part of the thesis, we saw that PCSK9 has a small effect on chemokine receptors, and maybe this is a small hint into its role apart from cholesterol regulation. So further studies are needed to discover what exactly is happening here. Now we looked at a protein that can affect receptors. For the next part of this thesis, we wanted to directly look at a receptor that can affect atherosclerosis. And for that, we chose calcium sensing receptor, or CaSR. Now, to understand again what calcium sensing receptor is known for in our body, imagine the busy restaurant kitchen again. At first, around 30 years ago, the scientists found few sensors here and there. They thought these sensors were just measuring temperature and controlling the air conditioning to keep things running smoothly. But then, surprisingly, the scientists discovered something later on, that these sensors are actually everywhere. They are constantly monitoring and adjusting all sorts of things. They can even overreact and cause little problem. So that's calcium sensing receptor for us. Originally, it was thought to sense calcium and therefore was named calcium sensing receptor. By sensing calcium, it was thought to keep the calcium levels just right in our body. But now, this receptor has been found to be present in all cells, or almost all cells, and can sense a lot of different things, and depending on which cell type it is present in, can cause different effects. Now, for understanding if calcium sensing receptor has a role in atherosclerosis, we took mice and we took out the calcium sensing receptor from the mice. So we had two different groups, one with calcium sensing receptor and one group of mice without calcium sensing receptor. And we fed these mice very fatty diet in order to have the atherosclerosis disease. And we compared the disease between the two groups. And we saw that there was no change in the disease. This was quite surprising for us, so we decided to dig deeper. Knowing that calcium sensing receptor has a different effect based on the cells it is present in, we decided to take out the calcium sensing receptor from the immune cells. So we again had one group with one group of mice with CaSR and one group of mice without CaSR in immune cells, and we fed these mice fatty diet to have the atherosclerosis. And if you remember from the first slides, we saw that these atherosclerotic plaques had a wall-like structure on top of them. And we saw that without calcium sensing receptor, these walls were thinner. So to understand why this happens, we wanted to look at the immune cells and see if they behave differently. So we took these mice, we took out the immune cells from the mice and grew them in the lab and saw that, and we were expecting that they were behaving differently, and as expected, they had reduced inflammation and they took up less fat in comparison to the ones with calcium sensing receptor. And they also had a high energy failure. Now, knowing that these cells are behaving differently, we were wondering, how do these cells lead to thinner walls? So we wanted to find out if these immune cells are actually destroying the walls. But surprisingly, that was not the case. And we saw that these cells are affecting the surrounding cells, which then leads to the builder cells in the arteries building thinner walls. So to conclude this project, we saw that the mice without immune cell CaSR had thin walls, which are likely or easy to break and can cause heart attacks or strokes. For the last and the final part of the thesis, we looked at fat tissue and atherosclerosis. In obesity, the fat tissue in our body enlarges and they secrete several detrimental effects that could lead to local as well as complete inflammation, which then could lead to atherosclerosis. There was a paper published that said that CaSR exaggerates these detrimental effects. And this was done in the laboratory conditions. So we wanted to see if this was true in mice as well. So using the same principle, we took out CaSR from the fat tissue, and we saw that there was no change in the disease. But when we took out the fat tissue itself from the mice, we saw that the mice without CaSR in the fat tissue had less cholesterol. So as a conclusion, even though there was no effect in the disease, the fat was somehow leaner. And to conclude the CaSR project, we saw that CaSR had a cell type specific effect, and it can play a role in atherosclerosis, and therefore these findings can be used to make CaSR as a new biomarker to identify the condition of the disease or as a new target to help overcome the disease. Finally, I would like to thank my lab, my institutes, and my university for giving me this opportunity, and my gratitude to my supervisors and the committee for your time and effort. And of course, this wouldn't have been possible without my friends and my family, and thank you all. I now give back the word to Victor.
Thank you very much, Ms. Candidate, for your very nice presentation, well illustrated, and I would like to compliment you already with this achievement, also that you took the time exactly within the 50 minutes. And before continuing, I would like to introduce your promotion team who guided you over the years, and I thought it's already quite a long time that you have been using this system. And the opposition team for the guests who are sitting here in front of us are sitting just opposite of me, and I would like to introduce from my side, on the right side, to Dr. van der Horst. He is involved in the immune lipid crosstalk at the so-called Maastricht Institute for Cardio-Renal Diseases. followed by, in the middle, by Professor Jankowski. He is head of the Department of Molecular Cardiovascular Systems in Aachen. And finally, on the left side for me here, all the way from you, the farthest away, is Professor Biese, and he is the chair in the Department of Experimental Vascular Pathology. The three of us have guided the candidate that she can now defend her thesis in the next 45 minutes. And for you, we have 5 opponents here today. One is online. She will be later on the screen. She is from Switzerland. She's now on that screen, but you also have to move back to see her. And we will start her, and she will be here at the end. So without further ado, we're going to start the opposition, and the opposition will be opened by Professor Schoen. She was also the chair of the assessment committee, and I would like to give her the word. She is officially professor in experimental cardiology at our university. And I'd like to give you the word.
Thank you, Mr. Prorector. Dear candidates, I would like to congratulate you on this very nice thesis, very structured, very well written, well organized, and also would like to involve your promotion team in this achievement. My background is in cardiology, so I have done my utmost best to come up with questions that are also related to your field of atherosclerosis. And there is some overlap, of course. It's all cardiovascular, as you said. So one of the things that I've also done early on in my career somewhere is a bone marrow transplantation experiment. And to start with a little bit, an easy question for you to answer probably, is can you explain to me, with respect to chapter 6, the difference between the bone marrow transplantation experiments that you describe for CaSR and the monocyte-specific knockout? Because they have different outcomes, and I was just wondering, what is the difference between the experiments? And why did you design those?
Highly esteemed opponent, first of all, thank you for the kind words, and thank you for the question. Indeed, that's a very good point, because you would think that we are looking at immune cells. So in the bone marrow transplantation mice, which is in figure 22, we had mice which were irradiated first to take out the immune cells from the bone marrow. And then we had the mice with the CaSR, full body CaSR knockout. And we we took out the bone marrow from these mice and flushed them and injected these into from the donor mice into the other mice, the irradiated mice, so that the hematopoietic cells are completely new. Mm-hmm. Whereas if you look at the monocyte mice or the macrophage mice, they had this CD115 marker in the monocyte macrophage, and we only knocked out the CaSR from monocyte alone. So in the bone marrow transplantation, you have other hematopoietic cells involved, but whereas in the monocyte macrophage knockout, it's just monocytes.
Okay, yeah. That's clear indeed. I was just wondering, because in the heart, when you irradiate a mouse and then you do a bone marrow transplantation, the tissue-resident macrophages tend not to disappear. Mm-hmm. So I was just wondering, so you only affect the bone marrow, basically. So did you check what happened in your atherosclerotic lesions?
Yes, so we did check the plaque sizes. We also checked the plaque composition in terms of a number of macrophages, number of smooth muscle cells. We did not see any change, which was surprising to us as well. Yes. But we did not see any change.
So could that lack of change be caused by the fact that you didn't actually get rid of the CaSR in the atherosclerotic vessel wall?
It definitely could be. It definitely could be,
okay. Yeah. That's interesting. And that also would explain, indeed, that in your tamoxifen-induced knockout, where you also hit probably the vessel wall macrophages, that you there do see a phenotype. Would that also mean that but I don't know anything about atherosclerosis in that sense. What is the role of tissue-resident macrophages as opposed to invading macrophages, monocyte macrophages?
Well, the tissue-resident macrophages, it depends, so the invading macrophages are most likely starting when there is inflammation. They come there with cues from endothelial cells and other chemokine receptors, and they are most likely to take up oxalveol and transform into foam cells as well. So I would say that invading macrophages are what we see in here. That's why the CD115 knockout had more of an effect than just the bone marrow transplantation.
Okay.
Yeah.
And would these invading macrophages, because you studied them in vitro in quite an artificial setting, and you conclude they are more M2-like, what would they be in vivo? What cues do they have to differentiate?
So, sorry if I'm understanding your question right. If CaSR is knocked out, what cues do they get?
Yes, well, in general, but then the next question would indeed be, what would the effect of lack of CaSR be? Yeah.
So, in the absence of CaSR, there are different pathways that this, especially in macrophages, CaSR is known for. For example, MAPK, ERK pathways that we also see in the kinase array in figure number 26 in page 194. There are a lot of different pathways that were downregulated, and we saw that these macrophages were less inflammatory as well. So there were a lot of different cues that CaSR was involved in order to cause this effect.
So now we are talking about figure 26. That's a good transition, because I was wondering what kind of cue did you give the macrophages here?
We stimulated them with LPS, lipopolysaccharide. Yeah, we gave them 6 hours of inflammatory condition, and then we tested them on actually all the figures from 25, 26, all of these were based on after inflammatory stimulus.
Mm-hmm. Yeah, I see that all of the kinases are actually downregulated. How did you quantify this?
So we have this very nice kinase assay. In this, we actually there is a chip with a lot of peptides, and that are downstream to the kinases that we want to look at. And we take the macrophages, lyse the macrophages, and put the cell debris on them. And then we saw that the kinases, the peptides that do not get phosphorylated or get phosphorylated significantly less than the control mice or the control macrophages. And that's how we can quantify this.
Okay, yeah. It's just that it's like a very blue figure. So I was just wondering about it.
Yeah.
Well, thank you. I've learned a lot, and I'd like to give back the word to the prorector.
Thank you very much. Then the opposition will be continued by Professor Schalkwijk. He was also a member of your assessment committee, and first of all, I would like to thank him that he is today our official secretary at this ceremony, and he accepted this task. He is professor in experimental internal medicine, and I'd like to give him the opportunity to discuss with you.
Thank you, Mr. Prorector. Dear candidate, first of all, of course, also from my side, my congratulations with this excellent work. And of course, with you also, the promotion team. I really like the work. It was very well described, very nice experiments, timely and highly relevant. So once again, my compliments. Where you have studied actually 2 enzymes in relation to cardiovascular disease. So in chapter 3, there was a focus on PCSK9 in relation to cardiovascular disease. And I was just wondering, you explained it very nicely in the introduction, the working mechanism of PCSK9 in relation to cardiovascular disease, the degradation of the LDL receptor, increased levels of cholesterol, and because of that, an increased risk for cardiovascular disease, atherosclerosis, cardiovascular disease. So what are actually the arguments for you to study a new concept, a new underlying mechanism chemokine receptors induced by PCSK9.
Highly esteemed opponent, thank you for the kind words and the questions. That's actually a very, very relevant and interesting point. We wanted to study PCSK9 because until now we have seen that PCSK9's role in LDLR degradation is very well defined. But there are also other receptors that PCSK9 seems to target, and slowly there is research coming up that PCSK9 is involved in a lot of inflammatory pathways. No, we don't know, though, whether this is LDLR dependent or LDLR independent. But like I said, there are so many receptors that PCSK9 seems to target, like scavenger receptors, other LDLR-like receptors. And it's not in the scope of this thesis, but we also briefly looked at co-immunoprecipitation, and we saw that PCSK9 binds to chemokine receptor. So, which made us think that it's probably, PCSK9 is probably affecting the amounts of chemokine receptors as well. And maybe this is a way through which it is affecting inflammation and that we don't know of. So this was it's one of the first times that PCSK9 has been studied in terms of chemokine receptors. So we were quite interested to do that.
So it is no degradation, it is really the interaction between PCSK9 and the receptor.
We don't know that yet,
Okay.
because even with PCSK9 and LDL receptor, there is a binding between the EGFA domain and the PCSK9. So we wanted to see if there is something similar happening.
And actually, chemokine receptors, a family of 20 different receptors. You studied 4. And why? Are that the most important receptors, or is it just a choice, or what is the idea behind that?
The idea was to choose 4 receptors and look at them, but we wanted to choose the 4 receptors that are involved in leukocyte retention, leukocyte migration. So that's the reason why we chose these 4. Yeah.
Most probably the most important.
Yes.
Receptors, I guess. So, okay, actually you found a lot of null findings, especially in relation to the animal work. And I was just wondering, I mean, you found also that there was a kind of an effect, or let's say an effect of especially endothelial cells after a pre-incubation of these cells with LPS. So it seems that inflammation is very important. Although you didn't mention that in your thesis, in your chapter, the effect actually of LPS in combination with PCSK9 in relation to the expression of IL-6. Nevertheless, it seems that inflammation is very important.
Yeah.
Nevertheless, you did all the experiments under healthy conditions. In vitro, under healthy conditions, but especially in the mice under healthy conditions, without inflammation or whatsoever. Well, you did the experiments in chapter 5 and 6 with the other enzyme under conditions of atherosclerosis. So why not in this chapter studying PCSK9 in relation to, let's say, underlying mechanisms in relation to cardiovascular disease? Why not in relation to, let's say, atherosclerosis or whatsoever? So why not in ApoA deficient mice?
That's actually a very interesting point, and I have been asked this before.
Okay.
So what we wanted to see was the initiation of the inflammation. And this is why we chose the Black 6 mice with 1 week of PCSK9 injection, and we wanted to see how, because inflammation is, it's all over the steps of atherosclerosis, but it's the important part for the initiation. And this is why we wanted to first look at a healthy mice and inject them with PCSK9 and then see if there is first any change. But the other studies that is to follow up was definitely to look at deceased mice as well.
So what do you expect actually? So a repeat of the experiments in ApoA or whatsoever in relation to outcome?
First, I think the next step would be to repeat in LDLR knockout mice to see if the findings that we saw are correlating there, and also to see if the findings that we saw are LDLR independent or dependent. And then, of course, to look at ApoE knockout mice with high-fat diet, 4 weeks, 12 weeks. Maybe 4 weeks would be better because the initiation, we can look more into this and the leukocyte migration. So that was, that would have been my next steps.
Okay.
Yeah.
So maybe a final question. So a lot of null findings, actually. Okay. But then it is very important to have a kind of a positive control. Is the system, is the mechanism working? So have you a kind of a positive control in the animals with overexpression of PCSK9? Have you tested, for instance, levels of cholesterol or LDL receptor? So proving actually that the model is working.
Yes, actually, we did. So in the supplementary figure, we also checked at supplementary figure 1 of the chapter. We saw first the levels of PCSK9.
Yes, that's
that
clear.
was upregulated,
Yes.
for sure. And yes, we did see the cholesterol was upregulated as well. Yeah, it was not in this thesis, but we did.
In the thesis.
Yeah, but it was definitely the cholesterol was upregulated.
That is very nice to hear. Thank you so much.
Thank you very much. Then the opposition will be continued by Dr. Kroon. Dr. Kroon is affiliated with the Department of Experimental Vascular Medicine at the Amsterdam University Medical Center. And of course, I first of all would like to thank him that he took the time to take the train and to come all the way from Amsterdam to join this defense. And I would like to give him the word.
Thank you, Mr. Prorector. Dear candidate, before I'm going to ask a few questions on your thesis, I would first like to congratulate you with the content of your thesis. And with those congratulations, I would also like to congratulate your promotional team. I actually learned a lot from your thesis, so thank you so much for that. And as you in chapter 2, you wrote a beautiful review on PCSK9 biology. So since you're the expert, I would like to ask you a few questions on that. So let's say, hypothetically, I would like to use the PCSK9 AAV injection instead of going for an LDL receptor knockout model, because it speeds up process significantly. I don't have to backcross on an LDL receptor knockout background. I don't have the expensive breeding steps. But based on everything you found in literature, what would you recommend? Would you recommend me using this model? Could you give a few advantages and disadvantages that I should keep into account?
Highly esteemed opponent, thank you for the compliments and the questions. Yes, that's definitely a usable model, but I think you're also asking this question because of what we found in the next chapter. So I have to say that the next chapter, we only did 1 week. So, of course, there was not enough time for the disease progression. And yes, you can use the model and for the instead of an LDLR knockout mice, but would be better to add a high-fat diet in order to make the disease to look at what we want.
Okay, so I'm glad to hear, because from your colleague, Professor Sluimer, I got some PCSK9 AAV, which we're going to inject soon. I'm really satisfied with these answers. Are there any particular mechanisms I should take into account when I give them a high-fat diet in terms of changes with LDL receptor knockout? Do I probably have difference in cholesterol levels? Is there something I should take into account in terms of the duration of the study or something else that I can pick your brains on?
So actually, what our peers actually did a project after this, and they did AV PCSK9 injection, and they did an obesity diet for similar amounts of time. So I think it would be relatable, and I think it can be reproduced easily. Yeah.
Great, thank you. Then I would like to move to, I think that's a nice bridge, I would like to move to chapter 5, talking about obesity. In your introduction, you described that it was shown that CaSR activation promotes pre-adipocyte differentiation, adipogenesis, while inhibiting lipolysis. And that was, I think, one of the rationales why you thought, okay, let's look into the role of CaSR in adipocytes, but then in mature ones. So my first question would be, does CaSR expression changes in terms of pre-adipocyte differentiation when, for example, compared to mature adipocytes, as you know, your findings were probably not what you have expected? Could you?
So the paper that you're referring to, the pre-adipocyte, they did it on the LS14 cells, and of course they are the pre-adipocytes, so they're not completely mature. And the ones that we did was directly the mouse knockout, and it is likely that the CaSR is better expressed in our cell model.
And is that known, or is that an assumption, just out of curiosity?
I am hypothesizing this because they already take out the pre-adipocytes and they start with the calcimimetics and calcilytics in that paper. So I am theorizing this. Yes.
Okay, thank you so much. So you measured also inflammation in your adipose tissue when you compared your wild type versus knockout mouse models, and those both models you gave a high fat diet. But did you also observe any differences in inflammation when you compare, for example, your chow mice versus high-fat diet? Because I'm just curious to see what is the level of inflammation you can actually reach in your adipose tissue. So do you have any insights on that? Do you have like a chow control?
Unfortunately, we did not do a chow control. We did the control with high-fat diet versus adipocyte CaSR knockout with high-fat diet as well. So, yeah.
Okay, thank you. So, in your discussion, you also mentioned the complexity of your adipose tissue. You mentioned it consists of many cell types, and I think that could be one of the clues to look into the role of CaSR in different cell types. So, and the interaction between mature cardiomyocytes and, for example, immune cells can play a role there. So, what type of like non-adipocyte cell types would you think could be important in driving adipocyte inflammation? Do you have any thoughts on that?
So if I get the opportunity to do this study again, I would definitely look into the perivascular adipose tissue as well. Apart from the visceral adipose tissue, I would look into perivascular adipose tissue.
And immune cells, endothelial cells, do you have any thoughts on that? Because you know those are also cell types that are either bridging or are next to the adipocytes, or immune cells that invade the adipose tissue. Could that be of any importance as well?
Yes, and we also actually looked at endothelial cells, especially specific CaSR knockout, and we also saw some interesting changes there. So definitely there will be interesting changes if we look at them cell by cell.
Great. So we have to look for the new PubMed hits soon, hopefully on that topic. Thank you. So I would also like to briefly. Do we have still time for a question, Mr. Prorector?
You still have 3 minutes.
3 minutes. Oh wow, that's good. So I would like to move to chapter 6. So in your CD115 myeloid knockout lineage, you see differences in triglyceride and cholesterol levels between your wild type and knockout mice. Right. So if I remember correctly, in your 4 weeks you saw changes in triglyceride levels quite profoundly, more or less like 40% when compared to wild type. In your 4-week mouse model, you saw predominantly changes in cholesterol levels. So, few questions. So, A, why is that? And how can like a calcium receptor in myeloid cells affect, you know, profound changes in lipid metabolism?
Yeah, that's actually a very interesting point, which we did not dig deeper into, to be honest. But I would say here that because of the CaSR knockout in the macrophages, the way they behave was completely different, and they were less inflammatory. Probably they were taking up less LDL, so the ox-LDL formation was also different, and this could be the reason why we see this. And also if you look at the 12-week high-fat diet, the cholesterol and the triglyceride levels almost comes back to normal, but here the disease is so exaggerated that it somehow evens out what we saw in the 4 weeks.
Yeah.
Yeah.
And did you also check the liver to see if you see changes in immune cell influx there that could, perhaps influence this lipid handling? Because, lipid uptake by immune cells could be one, but since you see quite, profound differences, do you think something at the level of the liver is happening? Did your supervisor or you look at the liver histology?
Unfortunately, we did not look at it. That's a good point for the next experiments. Yes.
Great, thank you. I think with that being said, I'm very satisfied with your answers, and I would like to give the word back to the pro-rector.
Thank you very much. Then the opposition will be continued by Professor Bot. She was also a member of your assessment committee, and she has a chair in cardiovascular pharmacology at the University of Leiden. And of course, I also have to thank her that she also took the time and has the time to join us today and to have some questions to you. And I'd like to give her the word.
Thank you, Mr. Pro-Rector. Dear candidate, I would also like to congratulate you with this very nice thesis. I've read it with great pleasure. I learned a lot as well. And also congratulations to your promotion team. I would like to continue the discussion you had with Professor Schalkwijk about chapter 3, but not before I asked one of your paranymphs to read proposition, they're not numbered, but it's number 6, and it starts with PCSK9.
One second. PCSK9 subtly modulates vascular inflammation via chemokine receptor expression, exerting effects beyond its canonical role in LDL receptor degradation. It's a quote from the dissertation.
Yeah, thank you. So this was already a bit discussed, that there are a lot of null findings in this chapter. So what data set makes you propose this statement? How can you defend this statement?
Yes, highly esteemed opponent, sorry. Thank you for the compliments and the question. Yes, so we did see a high inflammation in these mice. And when I said, you said that there was no findings, which is also what is in the chapter. And I explained to Professor Schalkwijk before that we wanted to look at a mice model that is not having an exaggerated lipid profile, not an exaggerated disease. So, which is why I can maybe not boldly say, but say that it's likely that there is some effect on inflammation using chemokine receptors.
Okay, thank you. So I would also like to ask a question about this model, because you just mentioned that you did see a slight effect on cholesterol. It's not in the paper, right? So I was wondering, because you have black 6 mice and you don't use a Western type diet, it's just a chow diet. I can imagine that this cholesterol effect is quite marginal if you compare it to mice on a Western type diet or LDL receptor knockout mice. So I was wondering about the time frame of the experiment, because now you have 1 week. Do you think you get a more strong effect on the inflammation if you would expand this experiment for a longer time?
Yes, definitely. So we even with the 1 week, actually the cholesterol levels were significantly higher. It was not even double, it was even triple, comparing the...
Even on a chow diet?
Even on a chow diet, just with the black 6 mice, but also the control on a chow diet and the PCSK9 injected mice on the chow diet. So we did see quite a high amount of cholesterol. So with an expanded timeline, it will definitely be much higher. Okay,
that would be interesting then to pursue maybe later on for your supervisor. So in your graphs, you show the chemokine receptor expression on the different subsets in the white blood cell population. Actually, I would like to go a step back. Did you see an effect on the white blood cell populations per se? Were these affected by the PCSK9 treatment? Like the levels of monocytes, levels of other white blood cells?
So what we saw is just the blood and the spleen leukocytes. So we did not, unfortunately, look into the
So you
white.
did not really assess whether the levels of the blood cells itself were affected by the
Oh,
treatment?
yes, we normalized we did not see any difference, and we
Okay.
normalized our findings. Sorry, I did not understand it
Okay.
earlier.
Oh, so you didn't see a difference there?
No, no,
So,
we
but
did
you
not.
do see this difference in TNF-alpha levels in the plasma of these mice.
Yeah.
So where does this come from? Do you have an idea? What causes this increase in TNF-alpha in these mice?
With the high cholesterol levels, I could say that this is through LDLR driven. This is dependent on LDLR, but we would need more studies to understand this.
Yeah, because you measure the chemokine receptor levels, but maybe the activation pattern of the white blood cells may be different, that they start to produce more TNF-alpha. Could that be an explanation?
Yes.
Okay, we'll continue with the in vitro experiments, because you did some in vitro experiments to test the effects of PCSK9, and you used mainly macrophages and endothelial cells. What made you choose these cell types for these chemokine receptors?
These chemokine receptors are very well expressed in these cell types, and we also wanted to look at them because they have an important role in leukocyte migration and retention, and this is why we did these in vitro experiments with these cells.
But why specifically the macrophages and the endothelial cells? Why? Because these chemokine receptors, like for instance CCR8 is mainly expressed on T cells. Why did you choose the macrophage and the endothelial cells for all these chemokine receptors? You didn't really, at least you didn't show it, have dedicated experiments where you have the specific cell types that would most highly express a certain chemokine receptor. Did you think about that?
Yeah, so our idea was to, like I said before, to look at the initiation of the inflammation, and in that the the endothelial dysfunction, and also the monocyte migration, and which is why we decided to focus on that, because they are the starting steps in atherosclerosis. So this was the reason why we chose these cell types.
Okay, and have you thought about like the monocytes or the neutrophils, for example, which highly express certain chemokines?
Yeah, we did not look into it, unfortunately, but we did think about it, and it would be the next steps to expand this further.
Yeah. So when you look at the endothelial cells in figure 13, there are actually some effects that you see. So you see increased CXCR4 expression, increased IL-6 expression. Is that related, do you think, that the effect via CXCR4 induces this IL-6 expression, or do you know the mechanism, what's happening? Do you have an hypothesis?
Well, with this data, we cannot really conclude that, but PCSK9 has been shown to activate a lot of different pathways that is involved in inflammation. For now, even recently papers have been published that PCSK9 can have an effect on TLR4, NF-kappa beta, and it could be one of the reasons, but to say that the CXCR4 expression is related to IL-6 expression would not be right at this moment without more data.
Yeah, I understand that. So maybe also in relation to one of the previous questions, you use LPS as a trigger in these cell culture experiments. But apart from this, you don't see really that much differences. Could it be that LPS, because that's a very strong pro-inflammatory signal, that it maybe dilutes all the effects of PCSK9?
Yes, definitely. It could saturate the effects of PCSK9, and which is why also we didn't see such effects. It definitely can be.
And could you think of a more mild inflammatory
I think I would
response?
add oxaldiol. Yeah.
Yeah, that would be a good suggestion too for future experiments. And then actually one of the final questions is, like, you now show this in early stages, you want to see the early inflammatory response, but actually in patients they have a prolonged inflammatory response. The patients are way older when they get this cardiovascular disease, maybe also due to PCSK9 mutations. So if you want to confirm your findings or maybe even expand your findings, if looking at patients, what kind of patients would you choose to investigate this specific question?
Maybe not the older patients first, and maybe looking at patients that have PCSK9 levels in a controlled manner, so as a control group, and maybe also patients that are already on statins or PCSK9 inhibitor. Maybe this was done as a preventive action, and we could perhaps look at these patients. And also younger patients to see if maybe PCSK9 levels are already higher, because PCSK9 is also an acute phase protein, because it can also be expressed more when inflammation or other diseases happen. So perhaps maybe we can take another disease model, and which can then lead to overexpression of PCSK9, and look at these patients in terms of inflammation.
Thank you. I'm very satisfied with your answers, and I would like to give the word back to Mr. Prorector.
Thank you very much. Then we move to the last opponent, and the last opponent will be online. She's already on the screen. The last opponent will be Professor Deuring. She's head of the Department of Biomedical Research at the University Hospital in Bern in Switzerland. And I also like to express our gratitude that she has found the time to join us online, and I'd like to give her the word. Professor Deuring, it's yours.
Thank you very much, Mr. Prorector. Dear candidate, I would also like to express my gratitude to be able to read that thesis. I really enjoyed. And I would also like to thank your committee and your supervisors and you for being inspired and staying in science, and then moving on. And with this, I would like to move to the questions I have. And my first question, I would actually like to come back to your mouse model with the PCSK9 AAV. I have read that AAV infection does impact differently on male and female mice. Can you say something about this?
Highly esteemed opponent, thank you for the compliments and the question. We did not separate the sexes to look for this, so I think without that knowledge, I think it would be hard to say if we had a difference between the male and the female mice.
Okay, so you did not differentially evaluate your data, nor did you do this. So because I think the female mice need a bit more AAV load in order to more or less reach the same knockdown of LDLR.
Yeah, we just looked at one group of mice with control and one group of mice with AAV PCSK9.
Mm-hmm. And in the adipocytes for the calcium receptor, you did not really see something in terms of atherosclerosis. What would be other cell types you would consider important? Is there any difference in terms of expression patterns of calcium receptor? And does it even matter how much? Or is there another type of regulation which is important in order to steer its function or modulate its function?
So in the adipocyte chapter, we did not see any effect, but we did see a slight effect in the cholesterol levels in 12-week high-fat diet. And also something that was interesting that we saw is a difference in the male and female here in the inflammation in the adipocyte, which is not in the scope of this thesis. And another type, a cell type to look at would be endothelial cells. We also did a little bit of experiments there, and we saw that the adhesion levels in the beginning of atherosclerosis was different when CaSR was knocked out. And also smooth muscle cells, these are all very important cell types to be studied upon.
Okay, but why would why would the receptor affect adhesion in endothelial cells?
Well, we did see that CaSR has a lot of effect. In the knockout of CaSR, we saw reduced VCAM, ICAM, and selectins. So why would CaSR have an effect on it? Because CaSR has lot of downstream signaling pathways that includes MAPK, ERK, and for this, the endothelial dysfunction is perhaps altered by CaSR, and therefore we see a different effect in adhesion as well.
Mm-hmm. And if you would need to think, let's say, ahead, could you already think about a therapeutic approach, which could be like could be kind of like taken from your findings, which could improve some kind of medication, for example, PCSK9 modification or statins? How could this be intermingled with your calcium receptor approach?
Yes, so for PCSK9 project, I would say with the findings that we have, maybe it's not the right time yet to use them in medication, because we need to dig deeper in here, but already PCSK9 inhibitors and siRNAs have shown to reduce cardiovascular disorders by a significant amount. Recently, there was a siRNA that was approved like 2 months ago, which was shown to have like almost 50% decrease. So in addition to statins, using PCSK9 inhibitors or siRNAs could definitely be an important thing to use in terms of cardiovascular risk. And when it comes to CaSR, with these findings, perhaps it's first to look at not just as a therapeutic agent, but as a biomarker in order to understand how the vulnerability of the plaque is, to see if the patients that have reduced CaSR expression in monocytes or macrophages, maybe they needed to be treated differently. And we have always been talking about personalized medicine, and these findings, I think, are definitely helpful, because we now see different effects based on the cell type that CaSR is present in. So this can be used therapeutically in different ways, depending on the patient's profile.
Okay, so you would then approach by a nanoparticle or something which would make sure it's cell-specific, the modification of the receptor?
Definitely a targeted way, and not as a broader range.
Mm-hmm.
Yeah.
And in terms of, I think you've already pointed out some of the limitations of your study, and I think there has been discussed a lot if the mouse is a good model and how much translation there is possible. What is your opinion on that topic?
Well, the mouse models that we use, especially the ApoE knockout models, don't show plaque rupture. They don't have the advanced lesions like in human, but however, the molecular mechanisms and the immune metabolism is very similar. So I would say the mouse models that we used are good in terms of understanding how the disease progresses, but maybe we need to look into other models, and I have read that rabbits or pigs can show plaque rupture. So those can be used if we want to study further into our monocyte paper. And for the adipose tissue, we can look into obesity models. So the mouse models are good enough, but we need better ones as well. We can look into better ones. Sorry.
Okay, thank you very much. I'm happy with your answers, and I'd like to give back to the Mr. Pro-Rector. Thank you very much.
Thank you very much. And we are just 1 minute. We are just missing 1 minute, but I'm sure that the beetle is on the way. So I know we have to wait. We can ask an additional question, and I just look if somebody of the committee would like to have an additional question to the candidate. If not, then we just wait for the beetle, and she will already joining us in any moment over there. There she is. Thank you very much. Dear Ms. Candidate, dear Ms. Sundararaman, the time appointed for defending your thesis has now officially passed. The degree committee will now withdraw. not so much to discuss the quality, that has been approved, but we're going to discuss the way you have defended your thesis, and we're also going to discuss the quality of your presentation. I request that you and your company await the results of our deliberation and the return to the aula. Thank you very much.
still standing?
Dear, I still call you, dear Miss Candidate, dear Miss Sundararaman, the degree committee here present has assessed the quality of your thesis, the way you presented your thesis, and also your presentation, which you have given this afternoon. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Biese is authorized to confer upon you this academic distinction in accordance to Dutch university customs, and therefore I invite your supervisor now to take the floor. Professor Biese.
Please all rise.
But first I have to ask you the following question. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay. Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Sai Sundararaman, I practice on this, the degree of a doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
So, first of all, congratulations, Dr. Sundararaman. So it's a real pleasure to say a few words today. As a doctorate, it's never a small task. And it requires patience, persistence, and more than a bit of resilience. So your PhD projects, like you already presented today, focus on this PCSK9 and the calcium sensing receptor. Now, those names might, before today, now mean a lot to most people here in the audience, but I think you convinced us it's really important players in biology and medicine. And understanding them better is really essential, not only for the science behind it, but also really as a potential for future therapies for patients. So of course, this path to this understanding was anything but straightforward. Science doesn't usually work that way. Some days experiments gave us clear results, exciting answers. Other days the data looked more confusing or simply refused to appear at all. And anyone who has spent time in a lab knows exactly how frustrating this can be. But you handled this with exactly the correct perspective. And you often reminded me, maybe also yourself a bit, that negative results are actually not negative. They're like we also said today, null outcomes. And they're still results. And they may sound simple, but I think it takes real maturity as a scientist to also accept this. And those no answers are also just as valuable as all of the yes answers, because they tell us where not to look and bring us still closer towards the truth. So, and then as everyday challenges of the PhD weren't enough, your journey came right up against the COVID restrictions. So the way we did science also changed, experiments had to pass, and travel came to a standstill. So it was a tough time for every researcher, but especially also for PhD students like you who were in the middle of their PhD projects. So, however, you adapted very well, and really kept your projects moving forward. So once in-person meetings and travel became possible again, you also made the most of it, and you presented your work also at the FCVB in Budapest or at the ES in Milan. And those weren't just the boxes to tick off at the PhD, but they were really moments to also share your science with others, to learn from others, and of course also to enjoy and have some fun there. So now you're continuing your career at Protembis, and I'm sure with your problem-solving mindset and resilience, you can make also a very significant impact there. And I'm glad that we're again working in the same building next to each other, and therefore still occasionally see each other. So last but not least, I also want to thank all the opposition members here today for taking the time for your defense, and also to challenge you with questions, and you were able to defend it very well. So it's highly appreciated that everybody joined. So Sai, congratulations again. Enjoy the celebration today, because I think it's a really a very great accomplishment that you can be part of. And I'm happy to give the word back to the prorector.
And we do a little applause.
Thank you.
So I'm not going to call you again Miss Candidate, but now I'm calling you officially Dear Dr. Sundararaman. How does it feel that?
Surreal.
Oh, real. Also, on behalf of Maastricht University, I would like to congratulate you with this academic distinction. It is in the Netherlands the highest academic distinction you can get, and you should be very proud of this, the way you have received it today in your day. In my congratulations, I also bring forward your promotion team, who brought you in the last years to this point, who designed the experiments with you, and you work with them together. I would also like to thank the members again, which also was done by one of your co-promoters, the members of the Corona who came here today from Amsterdam and from Leiden, and online from Bern in Switzerland. I always mention that you have 9 minutes, but it takes more than 9 minutes to prepare and to read the thesis and to digest the thesis as well. And my congratulations also your family here, always from, not all from India, some are from India, yes. And I was wondering about the city, but we're going to discuss this later at the coffee, because I wasn't there. And I would also like to thank them that they joined us today for this thesis defense. Now it's time to celebrate. And for the celebration, it will be as follows. I will officially first close the ceremony with my hammer. And then we're going to make a picture here in the aula. You don't have to wait for that. We're going to make a picture on the stairs of the university. But the audience can already go to the rafter, which is the place where you go outside to the right, and you will find there something to drink, something to digest, something to eat, and we will there in approximately join you in 7 minutes. Thank you very much, and I see you later. Perfect.
Congratulations. Very well done. That was great.
I was very nervous.
You were nervous?
Yes.
Nobody record.